we felt that it was very important, especially being here in the Bay Area, that if we're going to talk about the intersection of AI and neuroscience, that we include industry, companies, which are actually driving a lot of important discovery in this space. So our next speaker is Kaykas. He is Chief Exploration with a capital X, Chief Exploration Officer and Head of Neuroscience at Insitro. Insitro is a Bay Area company that was originally founded by Daphne Culler, who was a faculty member here in computer science. And they focus on drug discovery and development using data-driven techniques. So very excited to hear about what is happening at Insitro.
It's a pleasure to be here to be kind of an industry representative, and kind of the talks I've seen so far remind me I was actually at the Allen Institute many years ago before I went into industry, so it kind of definitely brings me back to thinking about kind of neuroscience and perception, and spent some time talking to Christoph Cook there, which is very interesting. So, but this is going to be a very different talk, really at the intersection of kind of applied machine learning to study of neurological disease. And I'll take you through some of the work that we've been doing at Insitro for the last about 4 or 5 years. So, I think we all really appreciate that neuroscience drug discovery is very hard. Brain, as we all know, is very complex. Orders of magnitude more synapses in the brain than the stars in the Milky Way. That's always kind of makes me dream a bit, ever since I was a kid. We really have a poor understanding of disease mechanism. I think there's beginning to be more understanding of disease mechanism, but that really is a challenge. There's really lack of predictive animal models for many of the diseases that we study. And that makes it very hard to identify targets and take drugs into the clinic. There's really a lack of biomarkers, and many of the diseases that we study are heterogeneous. We classify them as Alzheimer's, but it's there's many diseases there. We say this is Parkinson's, but there's actually many different kinds of diseases rolled into those. There's really difficulty in measuring the clinical outcomes, and there's kind of disease progresses very slowly, and so it's very hard to run clinical trials within this space. And there's really a high, this all leads to really a high rate of failure within neuroscience. So if you look across kind of different therapeutic areas, from phase 1 to the clinic, things like metabolic disease, kind of are quite kind of the probability of success is quite high. Kind of higher than just general indications. But if you look at, say, psychiatry or neurology, those are less than the average. This really plays out that the complexity of the brain and just our lack of understanding, plays out in our success rate in the clinic. And so at Insitro, we really, taken a step back and asked, can we use machine learning? Can we apply machine learning to, help with this problem? And it's, I think it's still a work in progress. It's going to be a many-year work in progress. But I think this is really something that we're kind of endeavoring on. So we've really focused on building kind of a machine learning first approach where kind of data is at the center. That's very different than most biotech companies or pharma companies. I was at Novartis previously to being at Insitro for 5 years, and it was very much kind of bespoke biology without the pipelines, without data was kind of siloed. So this is really a different approach that we're really trying to make data the center, and we focused on really 3 kind of data modalities that we've been capturing. So we've been capturing clinical data and bringing that in. We actually was able to form partnerships to get some of that clinical data. I've been really heading up the work on generating cellular data. And so, can we generate cellular data, with say iPS-derived neurons or other cell types to really help us try to understand, what goes wrong in disease. And then we focus on DNA and encoded libraries to generate data in chemistry space. But it's really, I think, the power of all of these, that we hope that we're going to be able to do, better biology discovery, really find the right targets for the right patients, develop, better drugs. And then really be able to move into the clinic. And we're just starting to go into the clinic this year, actually for metabolic disease. And it plays out, kind of neuroscience is hard, but we have actually programs in neuroscience that we're advancing. So the work that we've been doing on the cellular side, we've really been focusing on kind of the connection between genotype and phenotype. And I think most people are pretty familiar that targets that have genetic validation, things like PCSK9, kind of have a higher chance of success within the clinic. And so we've been really, both on the clinical side and on the cell side, really been using this and trying to connect kind of phenotypes to genotypes. And so we've very much kind of built this up and really started to build a map of genotype to phenotype within our cell systems to really understand kind of what goes wrong in disease. And so we've done this by focusing on using current tools. Everybody knows about CRISPR, but we've actually taken iPS cells or other cells and engineered them with CRISPR to make knockouts or suppression of many genes, diseases of interest for neurological disease or other disease. And then to phenotype those, most of the screens that have been done so far use things like selection in oncology. The cell's alive, the cells are dead, or you can use a flow cytometer and you can look at one marker. We've been focusing on things like single-cell RNA-seq or imaging, which have high-dimensional data, which are really good for kind of the machine learning problems that are out there. And so this took a bit of work to kind of build up the kind of imaging platform. I'm going to kind of take you through that. So we call this pooled optical screening or pooled optical screening in human cells. There's quite a few efforts out there, from the Broad or from Genentech or other companies are starting to actually do this. The way this works is you engineer cells with Cas9 and a barcode, and then you can actually do any kind of imaging on these cells, live cell imaging, or you could do, if it's a neuron, you can do calcium signaling, reporter assays, or you can do, live cell imaging. And then you fix the cells, and you add, kind of either antibodies if you want to look at particular proteins, or you can do something called cell paint, which I'll go through, which looks at kind of the overall morphology of the cell. Or you can do things like, adding fish probes to look at RNA. So this is kind of, you can basically do anything with this technology to look at, kind of, within a cell, what the features are. And then to kind of connect the phenotype to the genotype, we actually do something where we, in this case, we've got a guide RNA, and the guide RNA acts as a barcode. And then we actually amplify that guide RNA, and we do something called rolling circle amplification, and then we actually directly sequence that guide RNA within a cell. So it becomes kind of like a microscope, but a sequencer that's kind of combined. And you can see over here is kind of what it looks like when we go through cycles of reading out those base pairs of the barcodes, where it flashes different colors. And then you can kind of build back a connection between, okay, there's a perturbation in this cell that actually has a mutation, and okay, here's the phenotype within that cell. And so we've really focused on that as kind of a way to generate data at scale. And this process is very tedious, and so we've automated this. This is just an example of one of the automation systems that we've built. So this is kind of the machines part of my talk. And so kind of the sequencing really, each of those bases takes about an hour. It's very tedious. People in the lab who do this basically, over time they refuse to do it anymore because it's like 13 hours of sitting there, 1 hour, moving things back and forth. It's like the old days of PCR. So we've built up, kind of robots to do this, and we've really started to focus on building, up the ability to do kind of a, matrix of genes to features within cells, to really understand the complexity of cell biology. And more we use self-supervised models to look at cells. So humans are not great at looking at cells. There's like if you're a pathologist and you look at, say, oncology, over years you get good at looking at at the pathology. But in terms of cell biology, humans are pretty bad at looking at cells. You can't you look at a cell and you're like, it looks like a cell. Some people who spend lots of time can get better. And so we've been using things like Dino, which is from Facebook, which is a self-supervised model, to actually train the model to look at, okay, this is what a cell is, and actually instead of saying this is the nucleus, we actually use the model to actually define what the features are within a cell. And this is actually served to be pretty good for actually kind of understanding some fundamental aspects of cell biology. And so I'll take you through a few vignettes. One is just using this kind of approach to look at kind of networks of genes within cells. And this really helps us to kind of generate rich information about the pathways that are within cells. and gives us some foundation beyond kind of what's been published, throughout the years in literature. And then I'll go through a work that we've done on ALS, which is one of the main neuroscience projects that we work on, which is a really terrible disease. And we've really focused on using this to look at modifiers of one of the main drivers of ALS, which is TDP-43. So to kind of get this to work with cell paint, so cell paint is an assay developed by the Broad, by Ann Carpenter, and basically stains cells with different markers. And so you can look at things like the nucleus or the ER or the mitochondria. And it basically gives you kind of a way to featureize the cell and actually get information about what a cell is doing. It's kind of like RNA-seq, but kind of at the imaging level. And to do this, we modified the protocol to use a FISH probe because we had to be able to actually unstain the cells and then go through the in situ sequencing, which allows us to get the kind of barcode information that we need. And we started to kind of do this kind of at a small scale, something we call druggable genome, which is about 2,000 genes. And we've kind of walked our way to do this at a whole genome. And it's really been kind of time consuming in terms of the compute power and kind of the wet lab. So kind of have done this in a very much a kind of a stepwise way. And so this is kind of what you get when you take cell paint, which gives you the features of cells, and you use a kind of a pre-trained self-supervised model. We can either use models that have been trained on just random images, or if you use the model trained on some of data sets that we've generated and split the data, and then kind of have a test set and a training set, you can start to generate really interesting information where cells that look similarly, that have kind of genes, for instance, that are known to be in the same pathways cluster together, right? So this gives you kind of a map of just unbiased way of looking at, okay, these two genes actually affect this pathway, and the cells have a similar morphology. And so that's what you're kind of seeing here, which is all of these genes that are actually clustering into groups. And you can see things, like known things around, kind of RNA polymerase, or things like the TSC, mTOR pathway. And so it really gives you kind of just almost like a fundamental way of understanding, kind of cell biology without, kind of all of the data and the, 30 or 40 years of work that's gone into kind of understanding this. So to kind of understand, how good are we doing, kind of with these models, we use kind of a model trained on ImageNet, which just actually uses, kind of, any images from the internet, and we compared to, kind of, a small data set which has 300 genes, or this kind of bigger one that I showed, and kind of as you have, kind of the right data and more data, the models get better. And then we also wanted to know kind of the cell embeddings that we were using to generate these maps. Are they actually, kind of good at predicting kind of known biology? And so that's what you're seeing over here. So this is kind of a pooled optical screen derived network, with these different genes. And we compared it to kind of this database called StringDB, which has kind of all of these kind of networks, kind of over the last, 30 or 40 years that people have built up. And so we get a lot of the kind of known interactions, but then we get, additional new interactions, which are things that are, quite interesting to follow up of, new biology that one could look at. So we then scaled this up to genome scale to really, start to generate, kind of interesting maps on biology. And, what you start to get when you ask, self-supervised models to kind of then, cluster cells, you start to get kind of all these fundamental, pathway biology coming out. So like all the mitochondrial and ribosomal genes, or the cells that have those images clustered together. Things like heat shock proteins or unfolded protein response or ubiquitin ligases. There's just more and more data. And this is just on one kind of common cell line, right? So there's just tons of data in these images that allow you to understand biology. So in addition to kind of some of the known things, we were able to find some really interesting things around, kind of, within ALS. So for instance, TDP-43, and I'll go a little bit more into the program. TDP-43, or TARBP, is a protein that is found, mislocalized in 95% of all ALS patients. And if you ask, what are the genes that give you cells that are similar, you get a bunch of things that are, found in the nuclear pore complex, and there's really nice work from multiple groups basically saying that kind of nuclear pore is involved in ALS. So just another example of like how this data, just unbiased data, allows you to kind of understand fundamental biology. So summary of this part, we've generated really a platform to look at gene-gene interactions using cell morphology and using self-supervised models, kind of that can go beyond what humans can do to look at cells. And we've really kind of identified kind of known biology. That's the kind of thing whenever you embark on something new, you want to kind of, it's kind of like a control. You want the things that are known coming out, but we also are able to find kind of new things. And we really think that this kind of denser, if we do this at a much denser level, we're going to have some foundational information on the gene networks that actually regulate cell biology. So I'm going to take you through some of the work that we've done on ALS, really applying this platform, kind of this cell biology platform and imaging platform to look at can we identify kind of drivers of ALS, and can we identify genes that actually revert the phenotypes, at least in a in a dish that then we could think about taking forward into the clinic. This is actually partnered with Bristol Myers Squibb, so it's been nice to actually have their support, and they've got a big drug discovery engine that we can plug into as we actually make these kind of fundamental discoveries. And actually, it's not confidential. That was left from the it's all. So ALS is a pretty terrible disease. Really kind of people once they're diagnosed have 3 to 5 years to live, and there's really very few approved drugs. These are there's things that actually help are really not disease modifying, and they only increase the life of an individual for a few months. It's a fairly common rare disease. I mean, I have actually a family friend who was diagnosed with ALS, so it's the kind of thing where people know people that have ALS. It's not it's not ultra rare. And then 10% of all cases are caused by known kind of genetic mutations, and we've really used that in our studies. And then the majority are sporadic, and there's 25 known genetic drivers. All of or the majority of ALS has the same kind of pathology, where this protein TDP43 seems to be at the center of disease and becomes dysfunctional as people's disease progress. And so in this partnership with BMS, we generated a very large library of isogenic iPS lines where we actually made all of these 25 known mutations, and we used those to really understand the cell biology of ALS. And then we were able to take kind of technologies where you can take an iPS cell and kind of induce pluripotent stem cell, which you can generate from skin, and generate motor neurons from that skin, so that we can really try to study this disease in a dish, because it's very hard to get kind of biopsy from individuals. People don't want to give up parts of their motor cortex or their spinal cord unless they're dead. So we have taken this approach, and there's many people taking these kind of approaches with induced pluripotent stem cells. And then really key to Insitro, we've generated a lot of infrastructure and pipelines to capture all of this data. That's a critical part of what we do, is being able to just do this kind of work at scale. So in the initial work that we've done, we've really kind of focused on this gene TDP43, which I said becomes kind of mislocalized and aggregates in the cytoplasm of motor neurons, and really gets depleted from the nucleus. And more it seems like this gene, it's an RNA binding protein. It plays a role in splicing of many, many genes, 500 to 1,000 genes within motor neurons. And one of the genes called stathmin is involved in kind of regulation of axonal health. And so without stathmin, the axons degenerate. But there's just a plethora of other genes that TDP43 does. There's actually people moving into the clinic just with things that restore stathmin. So we really tried to take an approach where we're going to go upstream of stathmin and look at, can we actually find things that modify TDP43 and kind of correct its function at the genetic level. And so we used the kind of CRISPR screening platform and the imaging platform that I described to really model this disease in a dish, and then look for potential genes that could correct this phenotype when we knock them out. And so to do this, we generated a model, and we have a lot of genetic models, but in this case, we wanted a really strong model to have a strong signal and noise. So to kind of create a model for ALS in a dish, we depleted TDP-43 with an siRNA, and then measured Stathmin. So that's what you're looking at here. And if you deplete TDP-43, the level of Stathmin goes way down. You can measure that by a FISH probe. And then there's actually something called a cryptic exon, which actually is something that occurs during disease. And actually, people are starting to use those as a biomarker for disease. So we developed this. We then used our pooled optical screening platform to then do a screen on kind of initially kind of 1,900 genes, which are kind of, we call them the druggable genome, which are things that actually, there are some known already drug targets to those things, so that we could try to move fast. And this is just an example of the assay that was developed. So Stathmin, kind of is made as a pre-RNA, and we developed FISH probes to all of this. Then when TDP-43 is dysfunctional, you actually end up with it not being spliced correctly, because TDP-43 is actually involved in its splicing, and you get this kind of cryptic exon that's put in there, which is actually only seen in disease. And so then we actually developed FISH probes to that, and what you can see is when you deplete with TDP-43, the full-length Stathmin goes down, and the Stathmin cryptic exon goes up, and that's kind of what you're seeing here, and you can quantify that. So this is kind of a really nice kind of assay that has two readouts. We can also look at the kind of general morphology of the cell, which we've done with the kind of cell paint dye. And so when we did a screen for the genes, kind of that can correct this, what you see is a lot of the things that you'd expect, kind of come up. So if you directly kind of have a guide RNA against Stathmin, kind of the full-length Stathmin goes down. So that's kind of a nice, built-in control. Because we're using siRNA to kind of as the disease driver, AGO2 is actually involved in RNA processing, so it actually comes up as blocks this process. And then TDP-43 itself, because we're already knocking it down with SI, it doesn't really change the axis. So what you're seeing here is full-length Stathmin versus the cryptic exon. And so we're really interested in kind of these genes that are over in this quadrant. Those are things that kind of revert the phenotype and may be kind of corrective for patients. And these are things that exasperate, and it's interesting to look at a pathway level what these things do. So this is kind of very much like exploratory biology, where you're really trying to find novel things. So one of the genes that we're working on now with BMS, kind of this is a Western blot looking at if you have a non-targeting control versus a SI to the gene, it depletes that gene. If you then knock down TDP-43 to kind of drive disease, kind of Stathmin goes down. But if you then take this gene, Stathmin goes up. So this is the kind of thing that we would be looking at. And this is just also quantified, kind of with ELISA, with Western blots. We've actually, further, unfortunately, I would love to share the gene name. I hate doing this, but because it's in a partnership with BMS, we're kind of, that's the kind of thing where you kind of do. And we actually, in addition to kind of looking at just the Stathmin, we've actually looked at all the other genes that are downstream of TDP-43, and this kind of knockout of this gene seems to restore multiple genes, which is really a good thing, because you'd want to correct all of the defects and not just Stathmin. So kind of overall, kind of at Insitro, we've really focused on kind of the cell biology approach. We've got a lot of work on clinical data and bringing in kind of imaging and using that to drive genetic discovery also. But this has really been focused on building up this platform to look at cell biology at scale, using machine learning, using kind of human cells, and really trying to recapitulate as much as we can kind of disease in a dish, and then use machine learning to help us, guide us to find, kind of new insights. And we very much are kind of in the process of, having genes now. We're starting to work on BMS, on targets that target the gene that I described. It's kind of it's a complicated target in terms of drug discovery, but we're hoping to make, progress on that. And we've started to kind of do small molecule screening on that target to see if we can get kind of things that block its activity and kind of increase the levels of staph and all of the other proteins. Some of this work actually is published on bioRxiv now, so happy to point people to the paper. So it's nice to be in a place where we can publish some of the stuff and actually be a part of the community. That actually makes me kind of excited to be here, to be part of the community and actually get to share the work. Yeah, and just wanted to kind of thank the team. Insitro has been a fun adventure. I started, there was 10 people. Now we're about 300 people. Kind of all founded by Daphne Kohler, who was a kind of faculty member here in computer science. Yeah, so happy to talk to people about being in industry and the difference between kind of industry and academia, but have been in both. And yeah, just happy to be here and happy to take questions.
Great, thanks. Kong is chomping at the bit, so go ahead.
That's very exciting. So I assume that gene X, when you inactivate it, did not restore the TDP43 level.
Does not. Yeah.
Okay. And then what about, so you said that is also corrected other cryptic axon of other targets. That seems to be very exciting, suggesting that it's regulating splicing in a generic way. But does it actually have other wanted splicing phenotypes?
Good question. We have started to do some things. We look at global RNA-seq to ask kind of what does knocking out that gene kind of do. It seems that it doesn't do much unless TDP43 is depleted. So it seems like it seems to be more specifically correcting that. And we've been digging more into the biology, and there's a bunch of things now in the literature on kind of this pathway that it's regulating, which kind of makes sense with RNA biology. It probably has something to do with TDP43 binding to kind of the RNA and splicing it is what we think. But we're just kind of in the middle of really trying to figure that out. But yeah, that's a great question.
I have one, which is, so for myself, I'm a mechanical engineer that runs a robotics lab, and we don't really do, AI development in our research laboratory, but we're always trying to pull from whatever existing results there are in machine learning and such in order to utilize them. But it seems like every time we find a new method, a new technique, then something better comes out the next week. How as a company do you address, especially when you have so much infrastructure and investment surrounding a certain process, how do you adjust to the rapid changes in results in this field?
Great question. So, part of it is that we've tried to design a company that kind of locks versions and then kind of has a team. My role is head of neuroscience, but chief exploration officer is really to go out on the biology side and look for additional things that we could kind of pilot and see if they work, and then we would kind of add them into the platform in a next version. So we've tried to do that. It's a bit complicated, really. It sounds good, like you could actually do that, but then, as you say, you invest a lot in infrastructure, and then it's hard to change that infrastructure, kind of. So, I'd say this is kind of a work in progress. I don't have a good answer other than we're trying to kind of versionize things and actually, continue to explore new things, while delivering on the version 1 of the platform.
Great, thank you. So we'll go to Mackenzie.
Hi, Mackenzie Mathis, Swiss Federal Institute of Technology. I used to work on ALS stem cell derived neurons in a former life, it feels like, but I'm curious about how you guys are getting 14-day mature cultures from these human iPSC cells. So could you tell us a bit more about that?
Yeah, great question. So we've been using more of a forward program with transcription factors. The directed protocols are kind of where you use small molecules are quite slow and quite heterogeneous in the cells that they make. So we've focused on using protocols that were developed by Michael Ward, which use 3 transcription factors, and those surprisingly drive the cells to maturity. I mean, it's super fast. I mean, it's faster than I've seen any other protocols. And I've been in the field for a long time, doing all kinds of iPS-derived, stem cell, kind of protocols, even from my time when I was actually at the Allen Institute. Developed protocols there, all based on small molecules. And this protocol, within like a day, you have axons, and within kind of 4 or 5 days, they have all the markers for for motor neurons. Kind of whether they're whether it takes longer for the synapses to form, but still, like at day 10, you get, kind of pretty good activity, kind of when you measure in an NEA. So, I don't think they're perfect, but I think they're fit for purpose for our work. Yeah, but great question.
Thanks. Jay, next.
Yes, so just to contextualize my question, I'm a cognitive scientist who studied the effect of frontotemporal dementia on semantic cognition, and have been very curious about the etiology and the kinds of things that you're looking at in these diseases. And one of the things that's I wanted to follow up on was the fact that there's like 90% of the cases don't have the markers that you're talking about, and maybe to get you to say a little bit more about whether kinds of techniques and so on could somehow shed light on what's going on with those cases as well. Yeah. I guess you could get these cells from anybody who's got the symptomatology and then find out more about their particular individual cells ultimately. Is that part of the plan?
Yeah,
just curious.
Great question, and a really interesting, interesting question. So, for some of the genetic forms of ALS, for instance, kind of not FD, but ALS, like SOD1 does not give you TDP43, kind of pathology, and they're also FUS, which is another gene, another RNA binding protein, also doesn't give you the same TDP43 pathology. So we've been looking at those cells. We have genetic, forms of those that we're actually looking at, and we really want to see, like, are those really separate, or is there some pathway level, aspect, that would lead us to understand, kind of how these things must converge into, kind of, motor neuron death. I think the same would be true for, for FTD, in terms of, lots of shared genetics and lots of shared, pathology. So yeah, definitely don't have an answer, but it is something definitely that we're interested in.
Thank you.
Another question there.
Hi, I'm Tanvi Bharadwaj. This is more of a personal question. So I'm getting to the point where I'm deciding like what I want to study in college and what I want to do in the future. So I was wondering more about your educational and professional journey and the differences between working in the Allen Institute versus as an industry professional.
Yeah, great question. I'm happy to share. I mean, I did my PhD in virology, and then I did a postdoc in developmental biology at the University of Washington, and I actually moved to industry after that, kind of at a startup in Seattle that was spun out of a company called Immunex, and I was there for about 5 years, and then decided I had a joint appointment at the University of Washington, and then they were forming a new group at the Allen Institute, and really, I've kind of always been more, my role in the industry have always been more exploration and more, kind of forward thinking. I've been able to publish a lot in all the jobs I've had. I've been able to stay, kind of associated with industry. So that kind of moved back to, more of an academic role at the Allen Institute was kind of a nice, thing. It was almost like doing a second postdoc, and got to reinvigorate myself. But at the time I was at the Allen Institute, I kind of, then really realized I want to be in drug discovery. So that gave me an opportunity to kind of rethink. I think it's good in one's life to kind of have these opportunities to kind of explore a bit, and then figure out where you're really happy. A lot of people have a very straight path. Mine has been a little winding, and I'd say, that's, hopefully enriched me, as a person. So I think, now I went from the Allen Institute to Novartis, and kind of helped build a department there. Actually, the person who started that department was actually from Stanford, Ricardo Delmesch, who was here, working on stem cells. And so, I joined him. He was actually associated with Stanford and the Allen Institute, and so went into drug discovery. And for the last 12 years, I've been doing kind of neuroscience drug discovery. And but very much, I think everybody should explore and have time to figure out what you love. Like, I love what I do, and that's the most important thing to me.
Thanks for the great question and great answer. I think we might have time for one more question. If there's anything else. Yes, up here. Can we get a microphone?
Yes. I got it.
Over here. Oh, you got one. Great.
Hi, I'm Adriana Vela, founder of Market Tech Nexus. So I come from industry as well. So what am I doing here with all these massive brains. I love it. I love the science. I love to track. And my focus in business is to help companies implement AI, and not only that, but also the impact on humans from a brain science perspective. So I'm a leadership brain science coach, and I'm also a founder, and I'm also an AI consultant. That intersection is phenomenal. Yeah. So my question to you is, from a market perspective, where are we related to machine learnings and AI in general, to have some real output and make a real difference out there? Is it just still all very nascent research, or are we much more ahead in that curve? Any insights would be great.
My kind of, when I started at Insitro 6 years ago, I actually, I had heard about machine learning. I kind of tried to start doing things when I was at Novartis. In that time, things have really, in the 6 years, things have really, sped up, and there's, kind of a lot of interest. I think we're just at the start of all of this. I don't think we're anywhere near, kind of a place where, kind of it's solving all the problems and all the discoveries have been made. I think we're just at the start. And I'd say, like, in some of the research that I've done now, I can see, like, computer vision in the work that I do. Really, I can apply that. It actually does. It is useful. Kind of there are other areas where we've tried things that are just not there yet. It's there's so very specific problems, kind of that machine learning has been useful for. But I think over time it'll be useful for additional things.
Thank you very much. Let's give Aj a round of applause.